Hello, Mark here. Welcome to Lab 1 of the General Biology 2 course. Today's lab is going to be all about diffusion and osmosis, and what you're going to see in a little bit is me doing a simple diffusion experiment followed by a simple osmosis experiment. Before we get to that though, I just thought to give a little overview and reminder about diffusion and osmosis. Basically, you probably by now will have already had a theory class all about diffusion and osmosis from either Boris or Mackenzie, but this is just meant to get everybody on track before you watch me perform the experiments in the lab. So let's start with diffusion. Diffusion is basically a passive process by which solutes will go from a high to a low concentration to try and establish equilibrium. So this is passive. It doesn't require any energy from the cells or outside energy to drive this particular phenomenon. So for instance, if we were to take a beaker of water and some dye molecules in a dropper and drop a couple of drops of that dye into the beaker, what you would see initially is a concentrated spot where the dye has hit the surface of the water and kind of fallen down and started sinking into the beaker. If you were to leave this for a little bit of time, then what happens is the dye molecules will diffuse, and eventually, instead of just having a little small area of the beaker that has a very high concentration of dye, the dye will spread out to be equally distributed throughout the volume of the water. And this is basically diffusion. It's a passive process whereby molecules will go from a high concentration to a low concentration. So basically what they're trying to do is establish some form of equilibrium. One way you might have seen this, if you're a tea drinker, when you boil up a cup of water and you put a tea bag in, that initially you'll see that the teacup, the water is clear because the tea is still inside the tea bag. However, if you leave it to steep for a few minutes, then that's diffusion. The tea will leach from the tea bag from a high concentration within the tea bag to areas of low concentration within the cup until you have an equilibrium of a cup of tea ready to drink. Osmosis, on the other hand, is a form of diffusion. However, osmosis is the movement of water. This follows exactly the same rules as diffusion, whereby water will move from a high concentration of water molecules to a low concentration of water molecules. Now, this can sometimes seem a little bit confusing for students. Basically, it's depicted in this picture, whereby if we have a selectively permeable membrane, so we have a membrane here between the two sides of a chamber, and on one side we have a high concentration of free water molecules. So free just simply means that they're not associated with any solutes. So the solutes might be something like sodium chloride, might be something like glucose or something like that. The other side of the membrane, you can see that there are 1, 2, 3, 4, 5 molecules of solute, whatever it is. The number of water molecules is identical on both sides, 24 here and 24 in the chamber with the solute. However, the number of free water molecules is much, much lower in the side that has the solute, because what will happen is that water will associate with the solute particles and thus isn't free anymore. So instead we have 4 free water molecules on this side compared to 24 on this side. So what will happen is it'll move the water will move from a high free water concentration to a low free water concentration. So basically water will go to where the highest quantity of solute is. So what are the rules for diffusion through a real cell membrane? Because so far we've just seen examples of diffusion with dye and teabag, and also the movement of water through a semi-permeable membrane. Well, basically it all comes down to the properties of the phospholipid bilayer. Remember, there's these phosphate heads which are water-loving or hydrophilic. And sandwiched between them is a hydrophobic area that's made up of the lipid tails. And because of this, only certain things can diffuse through the membrane. It's still a high to low and a passive mechanism, but only certain things can pass through. And those things usually have to be very small, non-polar, and uncharged. Examples of this could be, for instance, oxygen. Oxygen in a high concentration, for instance in an alveolus, will pass through a membrane and into the blood to pass into a blood cell where oxygen might be at a low concentration, having already been around the body. So it'll pass by diffusion from the alveolus to the blood cell. So we have things like blood gases, oxygen and carbon dioxide, also nitric oxide, a signaling molecule, and so on and so forth. Other types of molecules and ions require help. They'll still be able to pass by diffusion from high to low, but they require a channel. So this might be an ion channel, for instance, and that ion channel could be a sodium channel. And if that channel opens and you have a high concentration of sodium outside the membrane compared to a low concentration of sodium inside the cell, then it will move down the concentration gradient through the open channel and into the cell. And you're going to be seeing more about this when you learn about action potentials later on in the course. Another example could be glucose. Glucose is a polar molecule, and it requires a transporter in order to be moved from one side of the membrane to the other. And when that transporter is active, perhaps by the actions of something like insulin, then again it's going to help move glucose from a high concentration to a low concentration. What we'll be using in the lab is a simulation of a phospholipid bilayer. It doesn't have transporters and it doesn't have ion channels. It's simply dialysis tubing. It's made out of cellulose and it has these little holes in it. And the rules for moving through the dialysis tubing, so this is going to be the first of the two experiments, are basically to do with the size of the molecules. So if we have something like this that's very large, and on one side of the membrane, high concentration on the left side and a low concentration on the right side, it cannot move through the holes in the dialysis tubing. So it's going to stay where it is. However, if you have another molecule within a solution like this, that's small and in high concentration here on the left and in low concentration here on the right, then because these molecules can pass through the membrane, through the holes, and there's a concentration gradient, then they will move from high to low, just like normal diffusion. And it doesn't matter if these are charged or polar molecules. As long as they're small enough to pass through, then they'll pass through and go down their concentration gradient from high to low. However, the large molecules will not be able to pass through because it's physics. They simply cannot fit through the little holes. So bear all that in mind, and coming up next is me in the lab doing the experiments. Cheers.
Hello and welcome to the Biology Lab. As you know, this semester's General Biology 2 course will be conducted completely online. So that means that unfortunately you won't be able to come into the lab. But what we thought we'd do is we'd have me come into the lab and film myself doing some of the experiments, and then you could see exactly what the experiments are, and then we could have a little bit of a discussion and maybe some questions about the results and the implications of the results so that you will understand what's going on. Today we have a simple diffusion experiment. I've got all my equipment ready here. Basically, I've got 4 tubes to start with. I also have some 15% glucose solution and 1% starch solution. I have 2 indicators, one called Benedict's, the other one called iodine or Lugol's iodine. And what we're going to do first is to actually establish what a positive and what a negative test look like for starch and for glucose. So what I'm going to do here is I'm going to take test tube number 1, and you have a little table that we sent you on Moodle for you to fill in as we go along what the results are. So the first one, if you follow along on the protocol that you were sent on Moodle, you'll see that I have to add about 1 ml of simple water. So this is just distilled water, nothing in it, and I'll add about 1 ml of that water to this tube. Okay? There. The second one, I'm going to add 1 ml of the starch solution. So be careful to use different pipettes for each one so that there's no contamination. Put that in there. Tube number 3, I'm going to again put water in, so roughly a 1 ml. And finally, in tube 4, I'm going to put 1 ml of the 15% glucose solution. Okay, so now I'm all set. So now what we want to see is what does a negative starch result look like. So if I add iodine, which is the test for starch, to the water tube, let's just see what the color ends up being. I'm going to add a few drops of this here, like that. Give it a little bit of a swirl. And I'll show these tubes close up afterwards as well. But you can see that a negative test for starch with Lugol's iodine is a kind of a yellowy amber color. So you can write that down in your table. Now let's see what a positive test for starch is. So this is the starch solution. So I'm going to do the same thing. I'm going to put 2 drops in there. And you can see that it's different to the negative test. You can see that a positive test for starch is this kind of very dark blue color with the Lugol's iodine. Now let's test the glucose. Let's see what a negative test for glucose is. So the first one, remember, I only put water in, and this is called Benedict's solution. You can see it's kind of a nice light blue color. I'm going to add that to the water, and as you can see, it doesn't do anything to the color of the Benedict solution. You can see it's still light blue. However, this particular indicator requires heat in order to be activated. So that isn't the final result. I'm going to have to put that into a water bath and heat it. So don't fill that in just yet. Here is the glucose. Put a few drops of Benedict's in there. Give it a swirl. You'll see that it stayed blue. But as I mentioned, this one requires heat activation. So what I'm going to do now is I'm going to transfer these two tubes to a water bath, and I'm going to heat them, and we'll see what the change in color was. Okay, so the tubes have been in the water bath, boiling for a little while, and you can see that there's been a color change. Let's just have a quick look. There's the negative control, the negative result for Benedict's, for glucose, so it remains this kind of bluey, nice light blue kind of color. And the positive glucose test with Benedict's, you can see it's this kind of muddy, greeny brown color. So in a second I'll show you a little bit closer so that you can record the results of the 4 controls into your table. So here is tube 1, which was the negative starch result. So it's this kind of yellowy amber color. Tube number 2, which is the positive starch color indication, you can see that it's dark blue. Tube number 3, the negative glucose test with Benedict's, you can see it remains kind of this nice blue color. And finally, test tube number 4, the positive test for glucose, you can see it's gone this kind of this browny, greeny kind of muddy color. So that's positive. Okay, so let's do our experiment. We've got our controls, and the second part, what we're going to do here is we're going to use some of this dialysis tubing. So this is like a semi-permeable membrane, and it's supposed to approximate a normal phospholipid bilayer. So basically it's dialysis tubing. It's used for people with kidney disease, so to help remove the waste from their blood and add back items such as bicarbonate and so on and so forth. And basically it has lots and lots of tiny little holes in it. So it's similar to a normal cell membrane, except obviously there's no ion channels here, there's no glucose transporters or anything like that. So basically what I'm going to do is to tie a nice knot in there, nice and tight. And what we're going to do is put a mixture of the 2 solutions that we tested earlier. So this solution here has 15% glucose and 1% starch in it. So I'm going to add that to the bag and then tie a knot to seal it into the bag. I'm just going to put that in there just to make it a bit easier. Take some of this. Just be careful to add it to the inside of the bag. There we are. Maybe add just a tiny amount more. Good. And what I'm going to do is I'm going to seal this end. I'm going to knot this end as well. Okay. This is the tricky part. It's worth taking a little bit of time to get this right because you want it to be nice and tightly sealed. There we go. Just tightly pull on that. Okay. So now we have the mixture of the solutions inside the bag, And what I'm going to do is I'm going to give this a bit of a rinse in distilled water that I've got here, just to wash any of that solution off the outside, just in case. And what I'm going to do is I'm going to be putting this tube, sealed tube with the 15% glucose and 1% starch inside the bag, into a measuring cylinder that has just distilled water in it. So distilled water has no solutes, so it's pure water. And then we're going to wait 30 minutes and we're going to see whether anything can pass through the semi-permeable membrane, because we'll be able to test the inside solution for both glucose and for starch, and we can also test the outside solution, the solution that was in the measuring cylinder itself, to see whether perhaps glucose passed through the semi-permeable membrane down its concentration gradient from high to low and into the outside solution, or perhaps starch was able to pass through the semi-permeable membrane down its concentration gradient from high to low. Perhaps both are able to, or maybe none of them do. So what I'm going to do now is I'm going to put my distilled water in here, I'm going to put the bag in there, and we're going to wait 30 minutes.
So here's just a little close-up of the bag. You can see that it's sealed, both ends, and the liquid, the mixture of the starch and glucose is on the inside, and I've knotted it so it's not going to come out. And now all we have to do is put the dialysis bag containing the solution into the measuring cylinder with the distilled water in it.
Okay, so we're all ready to go. The distilled water is in the measuring cylinder. We have our bag with the 15% glucose solution and starch, 1% starch solution inside the bag, and all we need to do now is wait 30 minutes, and then I can sample the inside and the outside of the membrane by taking some of the solution outside from the one that's actually in the measuring cylinder and take some from inside the bag, and then I can test for both starch and glucose and see what, if anything, is being able to pass through the membrane. Okay, so you're not going to have to wait 30 minutes because I can simply cut the video. Oh, and don't worry about him, he's armless. Right, so the 30 minutes are up. Here's our experiment. And now all we need to do is to sample both sides of the semi-permeable membrane. Remember that inside the bag we have a mixture of the starch and glucose. Outside the bag we have distilled water. And now we're going to try and see what, if anything, is moved from the inside to the outside of the bag. So what we'll do is we'll just take the bag out, the bag there like that. And I've got 4 tubes here. I've got inside the bag to test for starch, outside the bag, so from the flask, to test for starch, inside the bag to test for glucose, and outside the bag to test for glucose. So I'll just use, what I'll do is I'll start with the outside one. So I'll just take some of this and simply just add a little bit of it to a tube like that. So that's an outside. Here's the other outside tube. I'll take that one too. Put that to there. Just a little bit of there like that. Put that to one side. And now we have to sample inside the bag. So what I'm going to do is I'm just going to cut the top off like that. And should be able to, if I'm careful, to be able to get some of that out of there, put it in that one. There's some inside, and here's a bit more of that. Put that on the inside, so you just have to make sure to keep, not contaminate the other tubes. Just take that a little bit more in there. Finally. So now all I have to do is to add the iodine to the first two tubes to test for starch, and then add the Benedict solution to the second two tubes and warm them up in a water bath to test for glucose. So I'm going to do that, but I'm not going to show you the results. So what you'll have to do is to try and, using the information that you've been given and your knowledge about diffusion, try and hypothesize what you think the results would be, i.e. what the colors will be in each of these tubes, and thus whether glucose is able to have passed through the semi-permeable membrane from inside the bag to outside, or whether starch has been able to pass from inside to outside, whether neither of them have, or whether both of them have. So use your knowledge to try and figure that out, and those questions will be on Moodle for you. Another consideration is, what do you think happened to water? Given that you have distilled water on the outside of the membrane and a highly concentrated solution on the inside, what do you think happened to the water? Water can pass through the semi-permeable membrane, so that's a clue for you. So it either went into the bag or came out of the bag, and that'll be a question in Moodle as well, so be thinking about that too. Okay, so that concludes the first part of the experiment on diffusion. So now you can go to Moodle and answer the questions based on this experiment and see whether you understand the principles of diffusion as we've laid them out to you today. After that, don't forget there's a second video. We're going to be now looking at the effect of osmosis on red blood cells. So I'll be taking some of my blood and putting it into different solutions of sodium chloride, so different concentrations of sodium chloride, to see how that affects the shape and arrangement of the red blood cells. So stay tuned for that, and I'll see you in the next one. Cheers.